there's a lot of organisms that will live inside, say, whatever, Cliff Stevens Park, right? So we get back to this idea of fundamental niche versus realized niche. And, in the most general sense, you can think of an organism's niche. Actually, your book says it, and I really don't like it. An animal's niche is not its job, right? That's not its job. It has two different things. The niche first and most primary part of it is the physical conditions, the abiotic conditions where the species can survive, right? So where can we survive? And so this is going to be temperature, humidity, salinity. These are the physical characteristics that have to be. You're not going to find a dolphin in the desert of Arizona, right? It can't be there. So that's the first most fundamental thing. The next thing is the realized niche, and that's the spot where that it's a subset. So the first thing is a realized niche is going to be a subset of a habitat, and more importantly, even a subset of that fundamental niche. And that realized niche is the thing that will never be the same between species. Because if they were directly competing with each other, if two species had the exact same realized niche and they were competing, one would go extinct in that range. So the realized niche, you guys gave, some of you gave like a great example of like a tree. So there's a tree and there's like hundreds of beetles that could be living on a sequoia tree in California. They all have a different niche, a realized niche. Some of them are at the bottom. They like living in the dirt. Some of them living underneath the bark. I mean, it's so specific. Beetles are like crazy. So there's the range, a larger range. The fundamental niche is bigger than the realized niche. And there's actually ways you can model this. And I know some of you are out there like, oh no, no math. No, and it's okay. We won't do that. But you can actually model the fundamental niche and then the realized niche. And we'll talk about it a little bit because we do it for looking at where invasive species could go. So this fundamental niche is going to then dictate the geographic range, which is the area where a species can be found, right? We're not going to find dolphins in the desert. So that's one thing. So measure of the total area of where they could find, and that area is going to be defined by the conditions that they can use to survive. So this is showing you an example of what I was talking about. This is a teeny tiny little plant, Fremont's leather flower, that is found out in Missouri, and they're found in a specific area in these. So we already know they're found near rivers because they're found in glades. But if you actually look in these kind of swampy region areas, even within there, the individual plants have specific places where they want to live. So they are further restricted by variation in soil structure and in quality. And this, you can see this in the Everglades as well. I mean, the Everglades has areas that are wet, they have areas that are islands, areas on the islands that have high elevation, low elevation. All of this is going to dictate exactly where you find them. And you can actually look at different species, particularly related species, and try to figure out why do some species of plants exist right on the water's edge? Why do some of them exist in the middle of the island? In this case, they were looking at two little monkey flowers, which I don't think particularly look like monkeys, but I haven't seen one in the wild. So, monkey flower, that's fine. So Lewis's monkey flower lives at high elevations, and scarlet monkey flower lives at low elevations. So if you look at this diagram, they're probably both on the leeward or the windward side of a mountain range. One lives at the bottom, the other one lives at the top. And so what scientists did is they swapped out a few of the plants, because the idea was, are they living there because they have to live there, or are they living there because they can't outcompete each other? So when they were planted outside their natural elevations, they were able to grow, but they experienced lower survival. So in this particular case, these species are split out by the environmental conditions where they can actually survive. So they actually have a very slightly different fundamental niche. And there's a very narrow range in the middle here where both of them can survive. So as a general rule, populations are going to try to get bigger, and they're going to get bigger in an area that's more favorable to them. I don't think you don't find a whole lot of people living in Antarctica, right? You can't, can we? Yes, if we bring our own stuff, I guess we can. But there aren't as large populations down there. And so what a lot of ecological models will do is take these characteristics, like the amount of humidity they need, the water that they need, and they will mathematically place this over the planet to see where in the world could this species live if they were released, right? So that's the idea of ecological niche modeling, looking suitable conditions for them. This is used a lot in epidemiology. So when you're looking at diseases and how they spread, remember bacteria and viruses and protists, whatever, they're just organisms. They are just dictated by the same principles of ecology as everybody else. So you start looking around at, first of all, where are there conditions for them to persist and to be transmitted? For a big one for this is diseases that are mosquito borne. Where are there places where you're going to find mosquitoes, right? And so that's where do you have a location that people are coming in and out that they could transfer that disease from one spot to the other. And so you create this ecological envelope, these different envelopes on the planet. Just because that disease or that plant or that flower doesn't live there now, it most certainly could live there. And this helps people to understand where the plants could have lived, like their historic range, and also where they would live if they were transported there. So this is looking at niche modeling from a Chinese bush clover. It was brought to the United States to try to help control some erosion, particularly when people were like, they take down all the trees and plants and put up a farm. And then people didn't want to live there anymore. They used up the soil, or they wanted to make some more food for their cattle, or maybe there was an old mine. They pulled all the copper out of it and they just left it, and all the mud and goop and stuff would flow away. And not surprising, we'll see this over and over again. They bring it over and it kind of gets out of control. It comes with the best of intentions, but it really never works this way. And so they took all the data from where it can be found, this dark purple and dark blue over in Asia, and they put it onto the United States, looking at its known distribution and the places where it could live. And you can see somewhere just past the Mississippi River, you probably can't live much there. And then there are ranges where it's really, really moist here in the Pacific Northwest where they could live. And in the case of the bush clover, there are regions where they could live that they actually don't. So what that means to the modeler is that there is a missing piece. There's something, ecological factor that's limiting their distribution. And it could be something as simple as a pollinator. It could be a predator. So, and then trying to figure out, well, why didn't it spread to this area? Is it a predator? Is that something that we could then maybe bring to another part of the US and see if that would be helpful? Although another big source of invasive species are things that we brought into, like the love bugs that were brought in to eat the mosquitoes, except they don't. And now they just stick all over our cars. Now, the actual physical envelope where organisms can live is changing, and we've mentioned this a couple of times. We're already starting to see this in the North Sea and the fish populations there, because as the temperature of the planet warms, the water is going to warm, and that's going to expand the geographical range where many fish can live because the water is warmer. A lot of fish are limited by cold water, and so they just kind of stay where it's warm. And so looking here at the number of fish species collected in the North Sea, it has increased as the temperature increases. So now you have fish that used to live a little bit further south where the water was a tiny bit warmer, and they're able to expand their range up into the North Sea. So warmer northern temperatures have these southern fish expanding their range northward, and that's going to bring out some competition between these fish species. There will be organisms from the south that have the same general role as a mesopredator in their environment. When they move to the North Sea, they're still going to do that. So which mesopredator wins? Well, we'll find out.
So when talking about niche, is that going to be the bare minimum that they can survive, or is it going to be where it would thrive?
Good question. The bare minimum where they can survive, as long as they have enough energy to reproduce. But yeah.
Okay.
Yeah. So there are edges of your niche that where humans can survive, but it's like not the sweet spot. Now, the only reason that you would then live on those bare minimum edges is if a predator comes in that is so awesome sauce that you can't compete, and so you have to live on the fringe. All right. So when we talk about populations, the populations themselves have some characteristic. One that they like to talk about is endemic. So that's a species that is super, super specialized to live in a very small area, like a single isolated location. You see this in things like islands, particularly species that are only found in that one particular area. And then you have the opposite, which are the cosmopolitan species that have really large ranges and live frickin' everywhere. So like, I don't know, roaches, something not so pleasant. But they can be found everywhere. One of the things we're really interested in with populations is how many there are. What is their abundance? So how many individuals in that population are living in this area? So they're talking about the total number of lizards on a mountain, but you can think about it as like how many squirrels live on the Clearwater campus of SBC. How many squirrels are there, right? So the total abundance of the population gives us an idea of how much the resources they're utilizing. Is a population thriving, or is it going to be on the brink of extinction? Because you're looking at how many critters are there. And it's a sweet spot. As we talk about populations, we'll get to this idea of carrying capacity. You can have a really big population that appears to be thriving that is also on the brink of extinction because it's getting to that tipping point where there won't be enough resources. But that's, oh, don't worry. I know you're excited right now. I can see it on your faces. We'll get to that. We'll definitely get to that. Okay, then the other thing we talk about is the density of the population. Because if you have a critter that has a really big geographic range, there could be a lot of organisms there, but they're not really densely populated. You don't see them very much because they're spread apart. So density is the measure of how spread apart each member of the species would be, the number of individuals per unit area. And this is really simple. This is the same that you would do for density, you know, atoms per unit volume. Well, in this case, it's not atoms, it's like a cute little bird, right? So here we're looking at a map of bird species and individual per census route here on the left and geographic area. And as you can see, this bird species is found over a wide range. It's apparently not found to Florida except for the edge of the panhandle. So yellow is where the species has been found, but those darker oranges are where you have a lot more individuals per census route. So the density of that bird species is greater where the dark orange is as opposed to the light yellow. So near the edges of the range, and this goes right back to what Sierra was talking about, the edges of the range are harsher, they are less ideal, and so you see the densities of the population decrease. So within an area, you look at the patterns of density, and this is called dispersion, the spacing of individuals with respect to one another within that geographic range. And this is different than dispersal, which is moving from one spot to the next. So dispersion refers to the current geographic range. And there's basically 3 ways you can do this. You can have a clustered distribution, where organisms are aggregated in discrete groups, like these little fungi up here. You can have an evenly spaced distribution. This is something that you see when organisms are defending territories, or remember we talked about the leks, where the birds just stand there and defend the territory. And then you have random dispersion, when the position of each individual is independent of the other ones and is common due to an environment that has a non-random environmental heterogeneity. So basically the most common distribution is going to be clustered. Why? Because everybody has resources that they need. You have population clusters of humans. You look at the light maps on the planet, we're clustered around water resources. People need to be able to drink fresh water, and water is often used in our industry. So it's very clustered distribution. These mushrooms are sitting on the dead things that they need to decompose. You're going to find pigeons clustered around statues. No, it's not a good example. However, birds are often clustered around resources that they need, whether it's food resources, nesting resources. So cluster distribution is very common. Evenly spaced, while it seems to make sense, you don't see that very often because resources are not evenly spaced. And you see some random, but not too much, because again, they have to be inside the fundamental niche, and that's not going to be evenly spaced everywhere. If you need a certain elevation or soil type, you're going to be stuck in those particular regions. Now, what if you want to move out? What if you decide, I don't want to live in Florida anymore. I want to live someplace cool. My sister did that. She grew up in Florida. She did her postdoc in Maine and she never came back. She's been in Maine, DC, and now she's in Michigan. She loves it. She just moves out. But what if you're an organism and you're like, hey, it's too crowded here. I want to go to another area. Well, that's called dispersal, the movement of individuals from one area to another. Now, this is distinct from migration because migration is like an oscillation from, like, think of the snowbirds that are going to start coming down here. So you have organisms that come back and forth. It's a very seasonal, predictable movement from between habitats. And so dispersal happens more when a population moves to a new location. A whole group of them just pick up and move. And they're heading out west. They're going to start a new life in the palm trees and go to the Caribbean, something like that. But this allows species to colonize areas that currently are outside their geographic range. Now they have to move to a new place that is within their fundamental niche. They have to move to a place where the conditions are habitable, but we know through ecological modeling, there's many places that could be within the fundamental niche that just might not be close by. Why would an organism leave? Well, one, if they're an area of high competition. We talked about the male swallows last time, and they're just trying to get an opportunity to find a mate and nest. Well, if there's so many people, they can't do that, or so many birds, they might move to a different area to reduce that competition. Or they might disperse because there's too many predators. So they're moving to a region that's within their fundamental niche, but there aren't as many predators living there. Those are just some reason. So when an organism moves, this is a little Dr. B special from a different book, it's called range expansion. And this can result naturally, or it happens when we introduce them to a region where they did not previously exist. This is associated, we just saw this in the North Sea, with changes in environmental conditions. So a lot of times what you'll see are active movement of juveniles or adults. You see a lot of dispersal in young male organisms. So the elephants and the killer whales have matriarchal societies where the females live together, but the young males have to go find their own way once they become reproductive age. So that's when they have to disperse. We see this in male bottlenose dolphin populations. A lot of our local populations, the females generally live in one area, but the males will disperse to a new region after that. Another way you can have dispersal is by piggybacking on your favorite human. We have done a really good job of aiding human-assisted dispersal, other organisms in different places. We're going to do a whole section on invasive species, but it's often harmful to bring a species from, say, Australia, the Australian box jelly, for example, over to the US because there's no predators for it here. The lionfish is another great example of this. There's going to be competition between invasive and native species, and a lot of times the native species aren't ready for that. They didn't evolve with that organism in their environment, and so it can actually really greatly alter the ecosystem. One example, I'm talking about toxic box jellies that can kill you in lionfish, or you could be a moth, just this little moth here. It doesn't have to be. I mean, like you say, oh, you should wear a fuzzy one, but yeah, it's really dangerous. This is a gypsy moth that was brought over to the United States. I forget what they were using them for, but the idea was not for them to escape. Guess what happened? They got out. This is the other thing. If you're ever sitting on a city council or you're the science advisor for somebody and their whole idea is like, no, but they'll never escape, just say no. Just say no. It's like saying you're never going to feed the gremlin after midnight. Someone's going to do it, right? So caterpillars escaped. They found a way up. These are teeny tiny. And the problem is caterpillars are super destructive. They eat nonstop. I think we'll talk about this later, but man, baby caterpillars, for example, 2 or 3 years ago, I had a very successful crop of Italian parsley, and a very trusting female swallowtail laid her eggs on them. 2 caterpillars took out all of my bushes of parsley in a day and a half. That's how much they eat. So imagine if you had several gypsy moths suddenly running around in the trees of our forests up in the Northeast. they're suddenly gone because they eat all the time. That's all they are. For some moths and butterflies, they are only a moth or a butterfly for 24 hours. The rest of their life they spend stuffing their little caterpillar pie hole with your plants. So this is showing the huge spread from being released and escaping in the 1900s to 1994, and then because they can fly, they have a lot easier time being dispersed. It's a lot harder to go from the Atlantic Ocean into the Great Lakes if you're a fish because you have to swim there and there's not a lot of water in West Virginia. But if it's an organism that can fly, they can carry themselves, and they've done a pretty good job of that along the way. Another big example of this is kudzu. Kudzu was brought in again to help with soil erosion and as cattle food, but now it's one of the most, it's on the federal noxious weed list. You didn't think there was a list for that, but they have a federal list for everything. And kudzu is very pretty. This is a picture of one here. However, this is what it does. It grows so fast, it grows over everything. This whole open area is covered in kudzu. It grows up and over the trees, and it just destroys any plant in its path, not by eating them, but by blocking the photosynthetic radiation that it needs. So the distribution of populations can actually be estimated, quantifying it, and this will be, this is one of your activities. So we can look at the density and number of organisms in a couple ways. One is to do a census. So where you try to count every individual in a population. This is really impractical. You're not going to get every individual in a population, but you can try. But you can conduct a lot of bird surveys. So you'll walk through a population of the area and count the individuals that you see. This is very common for birds. And you can do area and volume-based surveys. So if you look over here on the right-hand side, you'll see a famous white square quadrat. Looks like it's about a meter. So you head out into the landscape, you place your quadrat down, and you only count the organisms in that particular quadrat. And it's much easier to count species in a 1 by 1 meter quadrat than it is to try to count it on that entire prairie. You would be there all day. Now, when you're doing this at a higher level of science, you just don't throw down one quadrat and call it good. You actually will select several patches within that habitat and count the individuals in those quadrats. But if you take enough samples, it's possible to get an estimate of how many individuals are in that average sample. So quadrat surveys, very popular. Line transect surveys are another one. We do this in marine biology. And this is where you set out a piece of tape it's like a measuring tape, and you count all the individuals that are along that line. So here you can see that it looks like this isn't a standard line because there's not any algae on it, but they've taken a 100-meter piece of line, they laid it over the coral reef, and you have a diver that swims along the line and counts everything on that point. Line transect surveys work for things that don't move. You can do line transect surveys on land, but I guarantee you, you're almost never going to see a squirrel standing exactly on that line transect, exactly where you need to sample. So it's very useful for benthic surveys, seagrass surveys, and for things like sometimes like crabs and lobsters, they don't move that fast. You can actually see them or they're likely to hide. So line transect surveys are utilized a lot in benthic areas.
So how could you apply that to a whole area, like a whole ecosystem? Because with the quadrates, couldn't you just, since it's 1 by 1 meters and the whole area you're measuring is like 100 meters, you could just multiply it, whatever you find, by 100. So like, how would you do anything like that with this type of survey?
So it would depend on the area that you're looking at. But usually when you do a line transect survey, you'll somewhere in the protocol, it will say how far do you look out. So you can say, I'm going to go a 0.5 meter on each side of what I see on the survey. So you can get an idea. It's larger than a 1 by 1 meter, but you can get a designated area. And you also, and you'll also try, you can even see in this particular area, they don't all have these flat corals. So you try to make sure you have transects in each area. But that's a really good question. That's how you would do it. And I want to say the Christmas bird count, which I've never done, but I have students who are very into birding. You can actually, when you walk a line for a bird transect, you do the same thing. You say, well, I'm only going to go a meter overhead and side to side. And actually, if you practice it, you get really good at distance estimations. But yeah, you would definitely need to quantify it. And what's interesting about line transect surveys is there are some transect surveys that have been stapled in the ground and studied for decades. So it's just a standard survey line. And when I went to the University of Miami, we would head down to the Keys to one of their research centers, and they had a standard transect, and they would have the undergraduates snorkel that every semester. So it's like free data for them, right? They get undergraduates to do the diving, and then they get the data back. But then you can show how the reef had changed over 10 or 15, 20, 30 years, because it's the same line, and you would snorkel it over and over again.
Gotcha. Thank you.
You're welcome. Other ways that you can quantify individuals is using the mark recapture method, and I'll be making a video about this for you. But the animals are, mark recapture works pretty well if you have an organism that comes back to that area every year, say a migratory species, or a species that tends to just hang around in that area. So as you see here on the right, that is a horseshoe crab. And here in Florida, we do quite a bit of mark recaptures with horseshoe crabs. Horseshoe crabs are not tasty, things that you find on your local menu. However, they have properties in their blood that make them very good for developing vaccines and medical equipment because it coagulates on any bacteria or impurities that might be in there. So they are harvested not to eat, but actually for their blood. So they're studied using this mark recapture survey. If you haven't figured out what you're going to do for your project yet, there might still be hope. So you mark individuals in the population, you go out and you try to catch every roly-poly you can. You're laughing at me, but you can. You go out, you grab every beetle you can, every roly-poly you can, and you mark them with fingernail polish. And then you release everybody back out there. You didn't hurt them. This doesn't hurt the horseshoe crabs. You release them back out there, and then you come back in a specific amount of time, and you decide that based on your literature. Do you come back in the next week? Do you come back in 2 weeks, 3 weeks? And you look at how many of those roly-polies with fingernail polish you capture again. So you go back out and capture every roly-poly you can. How many of them have fingernail polish on them? And what you're looking for is a change in the number of roly-polies with fingernail polish in them. If you didn't get them all, right, you didn't catch them all, you're going to find some roly-polies that are unmarked. So you look at the initially captured individuals, and then when you come back, you look at how many of your second batch are still marked compared to the total number of individuals in that sample. And this enables you to estimate your population size. If you have most of your individuals didn't have fingernail polish on them, you know there's way more roly-polies out there than what you caught the first time. If you go out and most of your organisms are marked, you have a pretty good idea that these guys don't move around and you got everybody. So you can do this a couple of times and get an estimate of population size. And so this is the basic math behind it. And I will be, when I make my video, I'll be giving you guys a couple of different examples of how to solve for it. But it's not too hard. It's basically just algebraic manipulation. So you're generally solving for n, which provides an estimate of population size. But you can use it to study any number on that equation. But you're generally looking for population size, right? So, and so in times past, back when we used to be able to catch lizards, unlike now, think about how would you mark in a non-invasive way the campus lizards to study their dispersion. Can you guys think of a way? Anyone type in the chat? I already gave you fingernail polish, so you have to come up with something else. So it can't be invasive. You can't like do surgery on them and give them a little transponder. That's cheating. They do it with fish though, believe it or not. What else could you put on them that would really hurt them, but you could see it easily?
Maybe a piece of
A piece of tape, maybe?
thread.
Thread? Okay.
Yeah. To their
Get
tail.
tiny, tiny knots. Yep.
Yep.
Iron knot. Come on, one more person, be brave.
I don't know, would permanent marker hurt them?
You can pick non-toxic permanent markers. I think that would be another really good idea.
Like
Permanent
some
marker,
gray tag
probably.
marker.
Okay, yeah, so thread marker, you wouldn't wanna, like, for example, the big thing we put on the horseshoe crabs, you wouldn't wanna put on a lizard, because that would like punch through their stomach, and it would be heavy. You don't wanna mark an organism with something that's gonna burden them, because they might not be able to swim as well as they need to. Like, you know, sometimes with dolphins or the great white sharks, they put big satellite tags on their dorsal fins. Well, it's not that big of a deal if you weigh a couple 100 pounds, but you don't put a satellite tracker on a pinfish that's about the size of your hand, because it's about the size of the organism. Right? So yeah, so you could do thread, you could use marker. You could try, yeah, you could do a sticker if you were gonna do it. It probably wouldn't last if it rained too hard, but if you're just gonna do a study of like daily movements of them and you were coming back over a week, a sticker would work great. And this is all science you can just do in your backyard. You can impress your friends. Like, we don't care, Dr. B. No one cares about this. But we can use these things. We can use tags in a different way. You can use them locally for mark recapture, but we also use tags to quantify how far organisms go from where they were born. So this is looking at a condor, and you can see somebody, he doesn't look too happy about being labeled with number 50. I gotta tell you, I think he was hoping for a luckier number. But you will, in migrating species, it's very common to put a tag on them and then have your birds are great because they're called, well, birders. People, the Audubon Society love to look for birds, and it's super exciting when they find a tag. So you already have people out there looking for you for free. So you mark it with this number 50. Anybody out there for the Christmas survey, a local citizen might see this tag and turn it into you. We do it a lot with fish as well. Believe it or not, they just put little cattle tags on some of the larger fish. So the state will go out, catch and release a bunch of fish, but put a physical tag on them. And then if a fisherman catches that, whatever, trout, amberjack, tarpon, there's information on that tag that they can then call in to the state and let them know where they were able to catch them. They do this a lot with the leg bands. You can also see it with birds that will have teeny tiny little silver bracelets on their legs, and they do that to determine how far away they moved. However, with leg bands, you have to catch that bird again. It's not usually easy to see if you're just looking at the bird on the branch. But when you're trying to quantify how far do these organisms go, right? How far do we have to extend our protection for salmon or for tuna? You're looking at their lifetime dispersal distance from where it was born to kind of where it was reproduced, or in this case, where it was tagged. And this gives you an idea of how much area might need to be protected if you're trying to protect that species. Or maybe there was a range that you didn't see before, and now you know, hey, we need that. Now, some species are super, super good at dispersing, and this is looking at the starlings. So who here has seen the starlings, right? They're in those big, they're not really that big, but they're darker colors, and they fly around in the sky in those really, really cool patterns. Well, what they have found by putting leg bands on them is that these guys are really good at dispersing. So they went in the 1900s, late 1800s or the early 1900s, somebody brought starlings to the United States because they wanted to have, it was something really obscure, like all the birds of Shakespeare in their life so they could listen to them, because that seems like a really good reason to bring birds over. But anyway, starlings love it here. Absolutely flipping love it here. So just from their lowly beginnings here in the early 1900s, they basically covered the entire United States by 2003. Kind of amazing. But in the case of starlings, you'll see they have these solid blue areas where most of the organisms live, but starlings have individuals that are really just willing to go, just disperse. They're really good at it. And so they were able to jump 4,000 kilometers in 60 years until now they're pretty ubiquitous. You could consider them, if you will, cosmopolitan in the United States and also in Europe. So some species are really good at dispersing, and probably because they have a pretty broad fundamental niche. And so when they get to a new area, there's a really good chance they can find a place to be happy. They're not picky eaters. Now, there are a few general trends that we see in populations. Populations that have really high abundance also tend to have large geographic range. So this is looking at the maximum abundance of bird species and the size of the geographic range of the bird. And you can see, this is natural logs, so it's much bigger than just 1, 2, 3, 4, 5. The larger the population in general, the larger the size of the geographic range of the bird. If you don't have enough resources, you're not going to be able to have a big population. Although now that you've taken regressional analysis, I wonder what the R-squared value is for that, because there's a lot of variability in there. I think my advisor would have said his class, like, I think if I squint my eyes and tilt my head, I might see that trend. But overall, you're going to have a larger range if you have high abundance, because you need to have enough resources to support everybody. So this suggests that reducing the range of any population is going to reduce the size of that population. And this is important because as Florida starts to build more and more houses, and we're having a huge number of people from the Northeast move down to Florida, anytime you put in a housing development, you're going to create a barrier to dispersal, because not very many organisms know how to cross the road safely. And you're reducing the size of land that they can live in, and so you're going to reduce the size of that population. They just can't support it. There's not enough space. And how much area and the population density you see is also related to body size. So this is actually on the left-hand side would be the body mass of, say, a mouse. And then out here on body mass, log scale in grams to 10 would be something larger, like an elephant or something like that. But the smallest critters we have the highest population densities as opposed to a larger individual. So organisms that have bigger bodies live at lower densities, so they're going to want more space per individual than, say, a nest of mice or ants or something like that. Now, this idea of dispersal is kind of essential to ensure the survival of populations, because bad things happen in small locations and you can have individual extinction. So what limits each species is going to be different, but there is dispersal limitation where you don't see this organism. It looks like a suitable habitat. They could live here, but they can't get there because there's some kind of barrier. The barriers can be things like interstates and roads. It can be oceans. They could be deserts. If you're a frog or an amphibian, it could be they can't move over to the new pond because it's too far away, and they'll dry out before they get there. Now, every now and then you'll find a case where organisms can get across. The Hawaiian Islands were famous for that. Either animals floating on palm fronds until they got to the new island. Sometimes you'll have birds that get caught in big hurricanes or storms and they're kind of carried across to a new area. And then we have humans. We're really, really good at bringing organisms to new areas, as well as creating new barriers for dispersal. And some of the examples they have down here: pine trees being transplanted all over California, aquatic species being transported in the ballast water of ships. We'll be talking about that a little bit more. But most dispersed species do not establish viable populations. However, the ones that do tend to be a real problem. So one way you can help populations find a new way or a new place to live is by providing a habitat corridor. And this is a strip of favorable habitat located between two larger patches of habitat. And this helps them to go from one place to the other. So this is looking at, I believe this was is this the butterflies? Yes, this is the butterfly study. So they have different patches of habitat. They have a large rectangle. They have one that's shaped like a couple shaped like a plus sign, and then one that had two semi-large rectangles with a clearing in the middle. And they were able to track the butterflies from one spot to the other, and individuals were able to disperse if they were able to disperse in the plot that was connected, because there was a little clearing where they could get through where the birds didn't get them. So this is just an example with butterflies, but we see this in Florida because we have started to create underpasses under some of our roads. And these are safe green areas underneath the big highways that allow things like bears and panthers to pass underneath without getting run over. So the Suncoast Parkway has a couple of these, and the Everglades, the I-75 Alligator Alley was named Alligator Alley because you used to hit alligators a lot out there. Way back in the day before it was a 4-lane highway. But when they built that highway, they actually tracked some panthers and other organisms and created areas where you go up a little bit, but there's actually an underpass below, allowing the critters to get across without getting hit. So you can create habitat corridors. And this is becoming more and more prevalent in conservation because that's kind of all we have. The human population is getting bigger and bigger. They're going to need places to live, but we can do things to connect habitats to allow organisms to disperse from one place or the other. And that's one reason why you see protection efforts, particularly in this case, the Rio Grande that goes between the US and Mexico. They're working to push and protect some of those habitats because that allows the animal species to easily go from one side to the other. The Rio Grande is super huge. But for some organisms, the river is a very effective barrier. Another one that we have, and we'll talk about again later, are the Florida panthers. We're starting to create a big habitat corridor for panthers so that they can get from some of their regions down in the Everglades to some of the undeveloped areas on the inside of the state of Florida. And we're working hard as Florida starts to expand its road systems because as roads go through, again, that's a very effective barrier to panthers being able to go from one side to the other and find this area. But it's not just panthers and bears, because let's be fair, I'm all about protecting the environment, but I would flip and lose my mind if I woke up and there was a panther in my backyard. Okay. I don't know of a lot of people that was like, I love a bear going through my trash and scaring my dogs. I mean, that's definitely an issue when you have people and animals interacting. However, when you protect these habitat corridors, you're protecting habitat not just for panthers and bears, you're protecting habitat for less charismatic species, little critters that need that, birds, mice, even moving around, coyotes, foxes, things that need the protection but aren't as famous, if you will. So now we're going to talk about these organisms that do live in these distinct areas of habitat. And this is, when you have different patches of habitat, you would expect everybody to divide themselves out so that they have the best quality habitat possible. So everybody starts out in this high quality habitat. What is the line where they decide to move from a high quality habitat to the low quality habitat? And it's quantified in something called per capita benefit. At some point, there's so many people living in the high quality habitat that you can't have what you need. There's no parking. You can't have a backyard. There you can't find a quiet peace of mind. There's just roads everywhere. So people are willing to move to an area that might be less densely populated, even though it doesn't have all of the amenities, because they're able to get a slightly better or the same per capita benefit. Now, birds and mice and foxes are not looking for movie theaters and shopping plazas, but there are still different types of per capita benefit that they're looking for. And so this modeling of the ideal free distribution helps to predict when organisms might move from an area of a high quality habitat to a low quality habitat. And this has been studied a lot in stickleback fish. If you haven't taken evolution yet, you're going to be hearing a lot more about sticklebacks. They're like little evolutionary superheroes, much like our friends Darwin finches. But researchers did an experiment where they manipulated the amount of food on each side of the aquarium. And what they found is that the fish are very simple. The food is the example of a high quality habitat. And so within a few minutes, the fish will start distributing themselves across the aquarium so that the ratio was approximately 4:1. And if they changed the abundance of the food, the side that the fish were on also changed. It's not that you didn't find fish on the lower side. You certainly did. You can see the low food side always had fewer fish, but a few fish stayed there Because if they move to the high food side, now I've got to compete with 8 fish. If I stay over here on the low food side, all the Daphne are mine. Now, food is not the only factor that organisms use. In some cases, individuals may not know there's another habitat there. The sticklebacks had it easy. It wasn't an aquarium. They just swam to the other side. Not everybody's going to know that. And also remember, food and places to live are important, but that just is one part of how organisms reach fitness. So they also have to survive from predators. They have to have enough mates. There's a couple of things that go in there. So this is another example of this idea of the high ideal-free distribution, and specifically it's looking at different types of oak forest because this particular bird breeds in these forests, but one is considered better to the other. Downy oak forests generally produce 6 times as many caterpillars, and that's what the birds need to eat. However, there's only so much room in there, so you do see a certain number of birds that will go over to the home oak areas because there simply isn't, and there aren't enough nesting spaces and food in the downy oaks. We can break populations from large populations to subpopulations. So you break them up into different areas, particularly in isolated patches. And when you start breaking smaller populations up into isolated patches, we create what we call a meta population. So not everybody is living in exactly the same spot and it may not be connected, but they can actually start to act as if they are a single population. And when that happens, we call it a metapopulation. And when you think of metapopulations, you generally have this idea of a source or a sink. So you have a patch of habitat that's really, really good. And everybody, once it gets too crowded, they move from the source population into a lower quality habitat on the outside that is a sink population. So they move out there. It doesn't have all of the resources that you need, but you do have some of it. So they move from one to the other. And the conditions for a metapopulation to work are that there's suitable habitat that occurs in discrete patches. The largest populations still have some risk of extinction, so there is a reason to leave. The habitat patches can't be too far away. There has to be the possibility that there could be dispersal, and that the local population dynamics are not exactly synchronized. So in this particular case, this is talking about a core population and smaller satellite populations. So when we look at what's driving this, there's local and patch scale changes that these guys have, birth and death, and larger regional scales, dispersal, colonization, and climate shifting for them. When we talk about a metapopulation, it's considered to be colonized, a new area is colonized when individuals move from occupied patches into unoccupied patches. And these moderate levels of dispersal between one area or the other allow these patches to have a balance between colonization and then organisms in that new colony dying off, which we call extinction. So for metapopulations to continue, there has to be a balance between them, between source populations recolonizing empty habitats. And this has been quantified. I'm going to skip past this because I'm not going to ask these types of questions for this exam. But this shows you the rate of colonization and the rate of extinction. Once again, it's a trade-off, and there's a sweet spot in here. The proportion of patches you find that are going to be inhabited depend on how quickly they die in that patch, as opposed to how many critters come over to that patch. If everybody dies there, no one's going to want to live there. And the example your book gives you here are this is looking at frogs specifically. So they have a high quality lake. They have a very important mossy, moist habitat that a habitat corridor to a lower quality. Maybe they have a lot of predators, and then a higher quality unoccupied patch. So you can look at an entire landscape and try to devise ways that organisms can get from these high quality patches to other high quality patches or any patch at all. And we connect all of those things together to preserve habitat for the panthers, or in other cases for things like elephants tigers trying to get these larger predators that require a lot of area to survive, getting them from one place to the other, because the more humans there are, the more patchy the habitat's going to become. And we want to be able to preserve as many species as possible. All right, and that's it for us today. I am going to be putting together a mark recapture study for you guys. Let me go ahead and stop this. So that way you can get a couple practice calculations in. It's a really fast calculation, so you're definitely going to have a mark recapture on the exam, and you may have, because I can put pretty easily into a multiple choice style question, and then a second calculation. So.
Lizards around here have too much solar energy. They're really hard to catch.
Yes,
They're so bad.
they are. So my husband's favorite technique is to sneak up on them from behind. So he'll find one on a smaller tree, and then he just kind of comes in and grabs them from behind. If
I'm
trying
always
to just,
afraid.
yours is afraid?
No, I'm always afraid I'm gonna hurt him.
Oh yeah, so yeah, well he grew up in Florida catching lizards, so don't judge. But he actually cups his hand and goes around the other side, so he'll be using a cup, so he never actually pinches them down. The other thing he doesn't like to do is their tails break, because then they're less attractive to the ladies, and he doesn't want to. While he loves to catch them, he actually loves them, and he's like, "Oh, look, isn't he cute?" I'm like, "Sure, honey, that's another lizard today." But that's how he catches them. And he prefers to try to catch things like the brown and green anoles, because some of the skinks, he said, they bite really hard.
Yeah.
I don't worry about that because I don't catch lizards. But yeah, that's what I would say. And it is easier on a cold day. He does, even in the morning, even though it's pretty warm in Florida, that if you need lizards for something, like when he used to catch them for Comp Phys, and we'd have this bucket of like 8 lizards overnight. But that's his secret. And he taught his cousin Jimmy to catch lizards too. So yeah, that's a ton of fun. All right, anybody else? All right, well, don't be too excited, but look for that mark recapture video. I know it's going to be the highlight of your weekend. And if you want to go ahead and set up a Zoom to get together, go over your exam, just shoot me an email. That's the easiest way to do it. So I can look at the entire schedule. And yeah, that's it. Everyone take care. Bye.
Yeah.